Hello, this is the To Health With That Naturally Healthy in No Time podcast for big health topics taken in small bites. I'm your host, naturopathic Dr. Amy Newsom. This is Season 1, all about the MTHFR mutation.
This week, let's carry on with our fish oil kick and talk about gene expression, fish oil, and MTHFR. So fish oil, which we talked about last week as well, continues to be a big deal for people with the MTHFR mutation. Today, I want to discuss a couple of studies about gene expression, fish oil, and MTHFR, but first, I want to make sure that you understand the basics of gene expression. So when your body makes something out of your genes, it doesn't just read the DNA and make a protein. There's more to it than that. The process, however, can be broken down into two big chunks. Bear in mind that there's far more to it than this as well, but this will help you to understand the research we're going to talk about. Chunk 1 is called transcription. Transcription is the process in which DNA is opened like a zipper, and mRNA, which M stands for messenger, is made from one side of the zip, and the resulting mRNA molecule is then processed by your body. Chunk 2 is called translation, and translation happens when the mRNA molecule that you just made from your MTHFR DNA is used to make a protein, which in this case is the MTHFR enzyme. mRNA is genetic material, just like DNA, but the difference is that while DNA might be the ultimate blueprint, it's also giant, double-stranded, and not easy to work with. RNA of all types, including messenger RNA, is single-stranded and generally transcribed from that master DNA molecule. It's created in small segments, and it's the signal needed for your body to actually build the proteins that it's coding for. What this means is that mRNA is a good marker in a research situation, specifically this one about fish oil and MTHFR, for the action of the MTHFR gene. And it's one of the only ways that we can measure when the gene is doing something. So let's talk about gene expression in the homocysteine cycle with fish oil and MTHFR. So last week we mentioned that levels of fish oil and homocysteine were linked. The higher the fish oil intake for the person studied, the lower the homocysteine levels became. Let's expand on that. This study, published in Nutrition Journal, looks at the gene expression, meaning the mRNA levels, for different enzymes along the homocysteine pathway. This includes MTHFR, but also other enzymes, including MAT, CSE, SAHH, CBS, and MTR. I've made a diagram on the website to healthwiththat.com that will illustrate how this all fits in together because it's difficult to understand when I'm just talking about it. Human liver cells were treated with either DHA, EPA, or ALA, and those are all different types of omega-3 fatty acids, for 48 hours. A control group with no treatment was also kept for 48 hours and then studied. After that time, mRNA levels were measured for each enzyme in question. It was found that MTHFR was upregulated by both DHA and ALA. MAT was downregulated by all three treatment groups, but most by DHA. CSE was upregulated in the DHA and EPA groups, and no significant changes were shown in the other enzymes. This study is remarkable because it shows the action of MTHFR can be influenced with something as simple as an omega-3 fatty acid. The next study that we're going to talk about is even more remarkable. This one deals with pregnancy. Methylation is one of the primary drivers of a person's epigenetic state, and some of the most important methylation that will ever happen to you happened during your gestation. So research involving this period is especially important. This particular study, published in Biomedical Research International, was conducted on rats. Because previous research has shown that nutritional changes in the mother affect both polyunsaturated fatty acid metabolism and global methylation in the placenta, this study theorized that the changes are due to some regulation of the maternal enzymes in the methylation cycle by dietary nutrients. That was a mouthful. Essentially, what they're saying is mom's diet creates epigenetic changes to the methylation cycle. The study divided pregnant rats into 6 groups, and each group was fed a different diet. The control group, which is the group that they measure everything else against, got normal folic acid and B12 levels. There was also a normal folic acid and B12 deficient group, and then a normal folic acid B12 deficient that compensated with omega-3 fatty acids. Likewise, there was a high folic acid, normal B12 group, high folic acid B12 deficient, and high folic acid B12 deficient that compensated with omega-3 fatty acids. Placental mRNA levels were tested for MTHFR and a variety of other enzymes that would sound like alphabet soup if I read them out over a podcast, but you can see the full article at tohealthwiththat.com. Placental glutathione and phospholipid analysis was also performed. So in an effort not to bore your pants off, I'll get to the relevant details. As expected, the mRNA expression of MTHFR was decreased in both B12 deficient groups relative to the normal B12 competent groups. Interestingly, omega-3 fatty acids were able to return the mRNA to normal levels in the normal folic acid B12 deficient group, but not in the high folic acid B12 deficient group. This tracks with what we know about folic acid's double-edged effect on the MTHFR enzyme. A small or normal amount is okay, and far better, of course, than no folate intake, which is what these rats would have had otherwise. But too much inhibits the MTHFR enzyme. Placental glutathione levels followed much the same pattern. Omega-3 fatty acids were able to correct the glutathione levels in the normal folic acid B12 deficient group, but not in the excess folic acid group. My theory about this is that glutathione manufacture is more difficult in the presence of imbalanced folate or B12, but that it increases in the most imbalanced group, which is excess folate and deficient B12, in an effort by the body to protect itself with glutathione buffers against increased oxidative stress. Phospholipid levels tested higher in both B12 deficient groups compared to the control group. And in both of these groups, fish oil, omega-3 supplementation, reduced them. This study is so remarkable because we're looking at the time period when epigenetics is at its most potent, and when the protective effects of omega-3 fatty acids could potentially alter the course of these pregnancies and also the life of those babies. While placental levels are being measured, these changes may have an impact on the developing fetus, and we kind of have to assume that they do. This means that even in the presence of a somewhat unbalanced diet, leading to unbalanced methylation, omega-3 fatty acids offer a regulating effect and may mitigate some of the worst of the consequences of that unbalanced diet. Any studies to do with pregnancy are pretty remarkable, and they're very rare in humans because pregnancy is a vulnerable time, obviously, and ethically it's very difficult to get approval for research in pregnant humans. But this study shows real promise in terms of a simple solution, fish oils, right, which is very cost-effective and very easy, to help women to truly balance that methylation even if their diet isn't completely balanced. Thanks for listening, and I do hope you'll join me in Genetic ROCKSTARS, our MTHFR community, which, by the way, is full of amazing, incredible people, and I'm really excited to get to know everybody. It's been a tremendous, tremendous journey.